Well, welcome to our continued discussion of methods. This is Chapter 5, Part 2, How Do We Study Structure? So let's start off with talking about cellular stains, the first variety of which is a cell body stain. Something like a Nissl stain, which would stain Nissl substance. Nissl substance are large granules that are associated with rough endoplasmic reticulum. This is really great for localizing cell bodies because rough ER are only found in cell bodies. So using a stain like Crescel violet, which is a Nissl stain, is going to be taken up by cell bodies and will stain them sort of like a purple color here, right? Crescel violet. This is an image of a Crescel violet stained brain, so you can see here the dark portions are locations of cell bodies. So we can infer things about cytoarchitecture or the structure of brain regions based on the cells that comprise them, exactly where the cell bodies are and how dense those cell bodies are. Something similar but kind of opposite is the Weigert stain, which will stain white matter. So you can visualize fiber tracks with the Weigert stain. You can visualize cell bodies with a Nissl stain. And we certainly cannot talk about cellular stains without talking about the Golgi method. So Golgi discovered this method and used silver nitrate to stain a sparse random set of neurons in great detail. So this process was discovered quite a while ago, right, almost 150 years ago, and allows the really intricate examination of individual cells. We really got a great idea from this technique about what cells are shaped like. An advantage, kind of an accidental advantage of this is it stains a random sparse set of neurons, so you can see with really great detail an individual neuron in a field. It won't show you all of the neurons that are there and how they're connected to one another necessarily, but you can get a really great idea of cellular morphology. This technique was refined by the renowned neuroscientist Robonika Hall, and in fact, this method is still used sometimes today. So electron microscopy, specifically scanning electron microscopy, this uses a microscope that provides three-dimensional information about the shape of a surface of a small object. So scanning electron microscopy is really great for seeing really fine detail of cellular structures. So the methods we've talked about tell us quite a bit about how neurons are shaped or where they're located, but they don't tell us much about connectivity. Tracing techniques allow us to see where neurons are going, which neurons are talking to which other neurons. So we can accomplish this in one of two ways. There exist retrograde tracers, which, like the name sounds like, these are substances that are taken up by terminals and transported back to cell bodies by cytoplasmic flow. So they move in a retrograde fashion, right? They're picked up by terminals, travel via exoplasmic or cytoplasmic flow, and arrive in the cell bodies, at which point we can visualize their presence. Anterograde tracers are just the opposite, right? Anterograde, moving in a forward direction. This is a substance that can be taken up by the cell bodies and transported back to the terminals by cytoplasmic flow. So they move in the opposite direction. They're taken up by cell bodies and travel down to the terminals. There are a number of different mechanisms by which we can conduct neuronal tracing, including both viral and molecular methods. So there's a number of different substances that we can use to do this that all accomplish basically the same end goal with differences in methodology and how we would visualize those things. So what's the point of having tracing techniques? Well, this allows us to sort of trace the projections from one brain region to another. So let's say, for example, you are curious about how these three brain structures are connected. You think that the ventral tegmental area, probably talks to the amygdala in some fashion. The amygdala probably talks to the prefrontal cortex in some fashion. So how can we verify that? Well, we can start by putting a retrograde tracer in the prefrontal cortex and seeing where it goes. So we drop some tracer into the prefrontal cortex. It's taken up and is transported backwards. And we sort of look through the brain. We take sections and examine this under a microscope and see where we can find that stain. And unsurprisingly, the prefrontal cortex is very richly connected to other regions of the brain. Lots of places talk to the prefrontal cortex. But let's say we take some sections from the amygdala and look under a microscope, and lo and behold, we find a bunch of our tracer being expressed within the amygdala. Then we can infer that there must be projections from the amygdala to the prefrontal cortex, right? We've used a retrograde tracer here, and it showed up here in the amygdala, which means that it's traveled backwards from the terminals in the prefrontal cortex to the cell bodies of the amygdala, which means the amygdala must send projections to the prefrontal cortex. Okay, so we've got. Next, why don't we drop some anterograde tracers into the ventral tegmental area and see where they go? So they're taken up by cell bodies, right? It's an interrograde tracer, so it's taken up by cell bodies in this region. And let's say we take some tissue and look, and lo and behold, we find a bunch of that tracer being expressed within the amygdala. So we can infer that there must be projections from the VTA to the amygdala, right? Because it's an interrograde tracer picked up by cell bodies here, transported forward to amygdala. So through years and years and years of different laboratories doing lots of fantastic work with tracing, we have a really great, albeit incomplete, map of how the brain is laid out, right? What regions talk to other regions through what sort of connections, oftentimes even with cell-specific specificity. So we might know exactly what type of neurons are talking to other types of neurons in given brain. So how do we visualize this stuff, right? Neurons are really small, and these tracers that we're putting into things are also really small. So what's a great way to amplify the signal of something that is that tiny? Well, the method that's often used is immunocytochemistry. So we take advantage of some of the innate properties of these cells and how our immune systems can interact with them. So just as a little reminder, antigens are proteins or peptides, such as those found on the surface of bacteria or viruses. These antigens are can serve as a binding site. Antibodies are proteins that are produced by white blood cells to destroy invading microorganisms. So when recognized antigens are present, antibodies will bind, right? So antigens are sort of like the binding site. Antibodies are these part of the immune response that can bind to antigens if they are present. So the immunocytochemical method relies on manipulation of this system. We're not going to go into great detail about exactly how, but molecular biologists have developed these methods to produce special antibodies that can bind to any given peptide or protein, right? So there have been antibodies developed that can bind to a wide range of targets. But these aren't ordinary antibodies. These are antibodies that are bound with something that helps us visualize them. So they could be radioactive, so they let off some sort of radioactive signal, or antibodies that are bound with a dye molecule, something that we can look at directly. That'll help us to indicate the presence of a particular protein or peptide. So the basic idea is you get a batch of these antibodies that have been engineered to bind to something specific. So maybe a receptor protein that you're trying to localize, see how many of a given receptor type exists. You get some of these antibodies that are going to identify and bind to those. And then you visualize that, right? So if you've got something like a dyed molecule, a fluorescent dye molecule perhaps, you collect the tissue, look under a microscope, and observe where you see that dye being expressed, right? So basically, the antibody is just a means to attach that dye to the protein of interest so you can then visualize it. So this figure is sort of just an illustration of those principles, right? So we have a protein of interest, then an antibody that binds to it. In this instance, we have a secondary antibody. Oftentimes, these reactions use a series of antibodies like this, where the primary antibody binds to these proteins, and then the secondary antibody carries a label. This is just a way of amplifying signal. This is a dyed molecule. In this instance, we can use a chemical reaction to produce a colored product, so we can visualize this very easily under a microscope. Here we have an immunofluorescence approach where the antibody is labeled with a fluorescent tag, which means that this tag will emit light of a specific wavelength when it is stimulated with light of a specific wavelength. So this is a nice way to visualize as well. So all of that stuff is well and good, but what if we want to visualize the structure of a living brain? Well, we have a couple methods that we can use to do that. The first is a CT scan, computerized. This is basically the use of a device that employs a computer to analyze data obtained by scanning beams of X-rays, producing two-dimensional pictures of slices throughout the body. So basically, this is just a collection of X-rays sort of laid on top of one another using a computer program to give you an image of soft tissue throughout. Next up is MRI, magnetic resonance imaging. This is a technique that we can use to visualize the interior of the body so we can see soft tissue with really, really nice detail. This basically involves interaction between radio waves and strong magnetic fields. This takes advantage of certain magnetic properties of soft tissue to be visualized. We also have the method of diffusion tensor imaging. This uses an MRI to reveal bundles of myelinated axons in a living human brain. So it can produce an image something like this. Here we have a visualization of projections between the thalamus and the cerebral cortex in a living human. So basically we use the movement of water molecules in bundles of white matter that are moving parallel with those axons. The MRI scanner is able to use information about the movement of water to determine the location and orientation of these axon bundles, giving us a more detailed map of connections within the brain. Okay, that concludes our discussion of studying the structure of the brain. Next time, we're going to get into trying to study the activity of a brain by stimulating and recording neural activity. See you next time.